live in 3, 2, 1. Just going to check out and see if my stream is working properly on Twitch and the music as well. Perfect, it is working. And in 3, 2, 1, action. Ladies and gentlemen, welcome to this science stream. We are going to be learning today about pharmacology medication. And the reason I wanted to do this is because I haven't done a science stream in a long time. And I just thought to myself, why not learn the subject of the day being learning about pharmacology respiratory medication. More specifically, the types of medication that are used, generally the types mainly that are used for people that have that suffer from like COPD disease or asthma or any type of obstructive pulmonary diseases. Anyways, let us get right into it and close off the year with our science stream. Without further ado, let us begin. The way this is going to work is that half of this presentation is going to be about essentially describing what each of these types do, and then we're going to go further into making a little drawing, perhaps, to better understand what goes on with this medication and into the body. So without further ado, let us begin with the preamble. So as I've stated and as I've said before, there is a wide variety of respiratory medication to choose from for patients suffering with respiratory pathologies, and I've stated them before. COPD, asthma. Asthma is technically considered a COPD sometimes, and others, you know, that could cause respiratory maladies or pathologies or etc. So, in this case, what it's going to help us, this is in order to perhaps help the people who are perhaps going through their school curriculum, and essentially it's to help the healthcare providers, in which they must know which medication is suitable for which patient, whether you're a doctor, a nurse, a respiratory therapist, or any other type of healthcare provider. That's what you must know. And also how each medication interacts with the patient depending on its type. And how are we going to do that? Well, first of all, by understanding each of the 3 types of respiratory medication, which are your beta-2 agonistics, your anticholinergic, and finally your corticosteroids. So, in which a broader understanding of these medications will enable the health provider to better be able to educate the patient whenever they have question, or to better conversate or better communicate with the doctor, the nurse, or the respiratory therapist, whatever healthcare provider you may be in the healthcare providing world. And so the thing that I thought would be best to better understand, and without just going through into this and just like basically launching you guys into the Shark Tank without knowing, I thought, why not have a little case study with our respiratory medication to better understand the different types of medication. So, in this case, our HPI, our patient here, starts with the HPI. Mr. John Doe, male, 64 years of age, has recently been diagnosed with COPD. COPD standing for chronic obstructive pulmonary disorder. He has been smoking since the age of 15. The patient does not want to stop smoking, even though he has received information regarding the negative impacts of smoking. The medications prescribed to him are tiotropium, otherwise the brand name known as Spiriva, or Spiriva, however you want to pronounce it, I don't really care. 18 micrograms once per day. Advair in discus form, 500 micrograms / 50 micrograms twice per day. It is a combination, and I'm going to get that to the combination shortly. And then our final medication is salbutamol in aerosol dosing form, or if you don't know what salbutamol is for you Americans, it's the same thing as albuterol. It's basically the same thing. It's just that in Canada we call it salbutamol. Aerosol doser form, meaning that it is an inhaler. 400 micrograms pro re nata, meaning only taken if needed, in case of, or whatever type of naming convention you may want to have. It is just in case for the patient, taken when needed for the patient. So, current medical history: patient has consulted the ER regarding a higher dyspnea than usual. The patient is coughing and his cough is hardly productive. Patient's expectorations are of greenish color and very thick. Patient does not feel well. Patient explains that he feels knife jabbing sensations on his left side. And so at the moment his vitals are his heart rate, 125 beats per minute. His blood pressure is 165 over 95 millimeters of mercury. His respiratory rate is 28 breaths per minute. His temperature is 39.5 degrees Celsius. And his oxygen saturation level is 85.0% at ambient air. So, with these vitals, we know our patient is tachycardic. He has hypertension. He is tachypneic or tachypnea, or he has a high respiratory rate. His respiratory rate is very high. And then finally, he is very, very incredibly desaturated. This is like red zone levels of desaturation, of deoxygenation levels. So we have to get this guy on some sort of either a 100% 100% mask or something, because this guy is desaturating a lot. And he also has COPD, so we could put him on also Venturi, because what our target saturation levels are, since he is COPD, he is known COPD, is 92%. Minimum. So, medical notes. Patient does not understand why he was prescribed such much medication, considering for him that they all do the same effects. He feels he is better off with the salbutamol only. Please take the time to explain to the patient how these medications act towards his respiratory system, which part of the ANS, otherwise known as the autonomic nervous system, is affected, the type of receptors involved, the intracellular action of the medication, and then finally explain why all these medications are necessary. Who? The doctor, the resident, or the attending has a lot of stuff we want to educate the patient on. So, let us decorticate, actually not decorticate, but disassemble the questions into multiple questions in order to better understand how to educate the patient. And the questions, to interpret them, we can interpret each question as how in the first part, how each of these medications act sorry, towards the respiratory system. I can't read. Which part of the autonomic nervous system, ANS, is affected, the types of receptors involved, the intracellular actions of the medication, and then finally, all of these, understanding all of these will finally permit us to better explain why all of these medications are necessary. And as such, the order of the slides are going to be as follows. The first part is going to be explaining the anticholinergic medication, which is our tiotropium, considered to be a double AC, otherwise known as LAC, however you want to pronounce it. Then it's going to further tell us the levels of the ANS affected. And then finally, the receptor and intracellular level. Same thing for the beta-2. The beta-2, which is our combination medication, and our salbutamol. Salbutamol, in this case, we're going to be mainly looking at the beta part, which is the which stands for beta-2 agonistic short-acting. I didn't include the, I included the other one, which is the beta-2 in the combinant, which you see right next to it. And then finally, the levels of ANS affected, the receptor and intracellular effects of the beta-2, and then finally we will finish off with the combination medication, which is a corticosteroid / beta-2. But since we're going to be already explaining the beta-2 in the middle part, we're just going to go straight ahead and jump into the corticosteroids. As the other one, the difference between the other one is that it's long-acting and the beta is a short-acting. Finally, the levels of ANS affected. Again, since it's a beta-2, we're going to also see that in the beta-2 section. And finally, the receptors and intracellular effects, mainly from the corticosteroid. So, without further ado, let us begin with our anticholinergic, which is our tiotropium, otherwise sold under the brand name Spiriva, or Spiriva. However you want to pronounce it, it does not matter. So, first, how does this medication act towards the respiratory system? Well, we know that this medication has a long-lasting effect in the lungs. It's an anticholinergic. So, the way it does, it acts by preventing the liaison between the ACH, otherwise known as acetylcholine. So, it prevents the liaison of the ACH substance and its respective receptors, ACH being secreted by the neurons, going towards two parts, nicotinic receptors or muscarinic receptors. We're going to get to that shortly. With that being said, it targets the smooth muscles, and that's what interests us from this respiratory medication, that are present in the lungs. Furthermore, this is a long-lasting anticholinergic medication, otherwise abbreviated as double AC or LAC, however you want to put it. And then finally, which part of the ANS is affected and which are the types of receptors involved. Now, you have a graph right below, and we're going to get to that really shortly. But first, I just want to explain that this medication will affect, first of all, the parasympathetic part of the ANS. So we know that if we look at the first graph, we have the ANS above, which has controls over the sympathetic autonomic nervous system, so the SANS. That doesn't interest us because it doesn't affect. It doesn't affect it. We don't care about that. What we do care about is the first path that goes towards the PANS, so otherwise known as the parasympathetic autonomic nervous system. And as such, inside of our parasympathetic autonomic nervous system, that's where we will find our cholinergic receptors, divided into two parts. First of all, the first part where you see the nicotinic receptors, also divided into two parts, but the nicotinic receptors are not found in the parasympathetic part. But it is very important, it's still very important to explain that pathway. So the first pathway, which is the nicotinic receptor, which divides into two pathways, like I said, ACH gets secreted from neuron to neuron, and then from neuron to muscles, which have nicotinic receptors. So the nicotinic, you're going to find them in two parts, between neurons and neurons, and then between neurons and muscles. That is considered, the nicotinic receptors are considered to be in the nervous system, which you can control. So they're not part of the parasympathetic, because you control actually the muscle contraction. You kind of control how the ACH is secreted. So, but that doesn't interest us, because you can't control muscle contractions in the lungs. What does interest us is the muscarinic, which is the second pathway, which will divide into 5 parts. And this is where it gets tricky, but here's where I'm going to make it as such that you can better understand what happens. So, the muscarinic receptors divide into 5 parts, M1 all the way to M5. So, if we start with M1, you will mainly find M1, and also the little pluses and minuses means stimulation. The plus stands for stimulation, and the minus stands for inhibition. So, with that being said, M1, if we start at M1, it will mainly stimulate, and you'll find M1 in the CNS and in the parietal cells. The parietal cells of our lungs, inside of the lungs, more specifically the bronchi and the bronchioles, bronchioli, and etc. So it's going to stimulate that. So since they're parietal cells, that does interest us. And also, wherever you have parietal cells, you also have blood vessels. So vasoconstriction. So that also interests us. So we're going to be more interested in that pathway. Now, if we move on to M2, the M2 inhibits, as you can see, there's a minus sign, but it only affects the heart. So that's not a really interesting pathway, because we don't care about that for our patient suffering with his COPD symptoms. So, moving on, we have M3. M3 is a stimulatory pathway, and mostly it's going to stimulate the glands, more specifically the exocrine glands, and the smooth muscle. The smooth muscle of what? In this case, we're targeting the lungs, so the smooth muscle of the lungs. It's going to stimulate what? Contraction. So that pathway interests us, and you're going to see why in a moment. Now, finally, M4 and M5, as you can see, it just diverges to the CNS. The M4 inhibits, while the M5 stimulates. Again, it's part of the CNS. That doesn't interest us, really, so we don't care about that. But if you notice, I will tell you of a. If you notice the pattern here, M1 all the way to M5. It starts with stimulation, then inhibition, stimulation, inhibition, and then finally stimulation. So if you kind of understood that pathway, you're halfway there. You halfway understood what these effects does. Now, we were talking about what the cholinergic receptors do. Now, if we add the word anticholinergic to it, we can now a little bit understand what these effects are going to be. If we add anticholinergic, the word anti to cholinergic, what is it going to do? It's going to prevent the cholinergic, so the acetylcholine, to do its effect. And we're going to see that a little bit when I will go into the visual details. Now, moving on to our beta-2 agonistic, which is our salbutamol. So, if we take again the question back from stage 1, how does this medication act towards our patient's respiratory system? Well, the first point is that this medication has a short-acting effect on the lungs. That's the effect of salbutamol. Or, again, for you Americans out there watching, you have what's called albuterol instead of salbutamol. It's the same exact medication. So, this medication will act as a stimulatory complement to the adrenaline or noradrenaline, or again, for you Americans out there watching, epinephrine or norepinephrine substance and its respective receptors. And we're going to get to the respective receptor shortly. Then, it targets the bronchi and bronchioles that are present in the lungs. And we're going to get that to its receptors because that's part of its receptors shortly. And then finally, this is a beta-2 agonistic short-acting medication. That's why we call it a beta medication, which stands for, again, beta-2 agonistic short-acting medication, as you can see for the acronym. So, second question, which part of the ANS is affected and which are the types of receptors involved? Well, the first answer is this medication will affect the sympathetic part of the ANS. So if I direct you down to the first half of the graph, you have again a graph presenting the two levels that the ANS controls. The ANS controls the parasympathetic and the sympathetic. The first pathway, which goes towards the parasympathetic, does not interest us. Again, because this is a sympathetic medication, which means that we are interested in the second pathway, the SANS pathway, the sympathetic autonomic nervous system. And as such, why? Because this is where our adrenergic receptors are found. So, adrenergic, meaning that they will accept secretion, the secreted adrenaline and noradrenaline by the neurons, that innervate the sympathetic nervous system. And now, technically, the adrenergic receptors go two ways. The first path is alpha, where you have alpha 1 and alpha 2 receptor. We're not gonna get into those because that's not where our main organs for our patient are. What interests us is the second pathway, the beta-2 pathway. Sorry, I don't know why I wrote beta-2 actually, I just noticed this. It should just be beta pathway, because then the beta pathway separates into 3 pathways. Beta-1, then we have beta-2, and then beta-3. So it should just be beta. I think that's a mistake. Sorry about that, if you've noticed it. And if you did notice it, thank you for calling me out on it. We are always glad to have corrections here. But anyways, so the first pathway that the beta has innervates or stimulates or acts as a complement to the adrenaline and noradrenaline that's secreted in the beta-1 receptors that are present in the heart and the kidneys. That's where you're going to find mostly beta-1. The second is beta 2, and beta 2, you're going to have a high concentration of beta 2 in the lungs. There's a lot of beta 2 in the lungs, and that's what our beta 2 agonist salbutamol, or albuterol, however you want to pronounce it, is going to affect. That's our main target. And then finally, beta 3, which are found in the liver and in the bladder. So, as you can see from this pathway, what interests us is beta 2. The adrenergic receptor that goes to the beta pathway and then goes straight to the beta 2 pathway. That's our main point of interest. And you're going to see why again in the drawing. And again, a little way to remember what beta 2 agonistic is that it's going to act as a complement. So meaning that it's going to stimulate what? For our patient, what do we want? We want that dilation, baby. We want the lungs to dilate, the bronchi in the lungs and the bronchioles to dilate as much as possible. So that's what we want for our medication. And then finally, we move on to our combinant, which is our Advair, otherwise known a combination of fluticasone. Fluticasone, we know is a steroid. Why? Because it ends with "-one", fluticasone. And finally, our salmeterol. Salmeterol is the second half of this medication, which is also a beta 2 agonistic, but this one is going to be long-acting. And as such, how does this medication, if we start back from question 1, how does this medication act towards our patient respiratory system? Well, the first half of this medication has a long-lasting effect. Well, actually, both medications have a long-lasting effect in the lungs, both the corticosteroid and the beta 2. So, it mainly acts as an inhibitor towards the secretion of pro-inflammatory substances in the lung. That's going to be the job of our corticosteroid. And then the beta 2 will target its respective receptors. I'm not going to get into how the beta 2 acts since I've already explained it in the previous slide. So we're going to jump straight to the corticosteroid part, whereas the corticosteroid will target pro-inflammatory secreting cells. And then the beta 2 half of this medication is a beta 2 agonistic long-acting, and that's the difference between our beta, and in this case, that's the acronym you want to learn, is the BALA. So, beta 2 agonistic long-acting, because what's going to happen is that this, the first half of this, the second half of this medication, which is your beta 2 agonist, it's going to take a little bit more time to act, longer time to act, but it's going to last longer versus the short-acting, which acts instantly for a bronchodilation, but has a short amount of time span. And then finally, the corticosteroid half of this medication is an anti-inflammatory, and we're going to get to that in a short while. So, which part of the ANS is affected and which are the types of receptors involved? If you've noticed at all, there's no graph. Why? Number one, because the second half, which is the salmeterol, if you don't know, is sold under the brand name Ceravent. It's sold under the brand name Ceravent. Think of snake, serpent, Ceravent. And it's the substance used in this combinate, which is our Advair, which is the combination of a fluticasone and the salmeterol. The fluticasone by itself, if you really want to know, is sold also under the brand name Flovent. But anyways, going back to the question, for the beta 2 half of this medication, please see the previous slide. I'm not going to go back into it and show you the graph of what it innervates. Again, just think of the salmeterol path part of this medication as a long-acting, whereas our salbutamol was a short-acting. So just think of it, just discern it that way. But for the fluticasone, so for the corticosteroid, there are no receptors involved for the corticosteroid have this medication. And first of all, since the steroid is lipocerebral, and second of all, we don't need a graph to explain that because there's no receptors involved for our inflammatory secreting cell. So now that we know the difference between all three, we are now able to give you a better representation of what each of the medication does. But before going towards that, I would just like to give out my references to my shout-outs to those who helped us or who helped me in this case, motivate me in order to do this presentation. I'd like to thank, first of all, Ninja Nerd Science. I think his name, the guy that does the videos for Ninja Nerd, is Zach Murphy. I think that's his name. Very cool guy. Thank you so much for your upload videos about the different pathologies, the pathophysiologies, and etc. It also motivates me to do these. And then finally, for the medication types and images, I'd like to thank the Réseau Québécois d'éducation en santé respiratoire with their medication, with their inhaled medication and their I'm looking for a word here. Their inhalation medication devices. That was the word I was looking for. So I'd like to thank them for that. Really do give them a check their. Please do check their channel or their website for better understanding. If you're interested in learning more, I highly recommend it that you do that. So, like I said, without further ado, to further understand our what the medication intracellular, to finally explain what the intracellular effects of the medication of each of the type of medication does, let us go finally to our drawing. So, since we had our anticholinergic medication, let's begin with that. And that's what I'm going to do now. And I'm going to have probably this guy right here with the square. So, but just before I begin, let me just show you the pathway that it takes that interests us, basically, before drawing. So, we have our ANS right here. Our ANS goes two pathways. We don't want our sympathetic. What we're interested in is this pathway over here. So, our parasympathetic nervous system. And then to our cholinergic receptors, the pathway, just a reminder what we want, the pathway that interests us for our anticholinergic medication is that we want to inhibit the stimulation of our M1 and M3 pathways. That's what we want to do. That target mostly the parietal cell, the exocrine glands, and the smooth muscle cell. So that's what we're mainly interested in. And that's what we want. So without further ado, let's begin with our drawing. Actually, I'm going to make it a little bit bigger. Oops, that's not my square. I think that's good enough. Hopefully that's enough space. So, as you can see, we're going to start off with my cell over here with the semi-permeable membrane. And what we're going to have here is our semi-permeable membrane. Whoopsie daisies. Yeah, OneNote is not the best, but it's going to permit me to do this wonderful drawing that I will now start. So, the borders of this is our semi-permeable membrane. And what do we have on our semi-permeable membrane? Well, we have our receptors. And what are they? There are cholinergic receptors. And what do you remember? What are they called, our cholinergic receptors? Well, they're called muscarinic. So, in this case, meaning muscarinic, meaning that they will accept ACH, which stands for acetylcholine. And acetylcholine, let's assume that our neuron secreted the acetylcholine. And what does the acetylcholine does? It's going to go attach itself to its receptor, to its muscarinic receptor. So this over here is a muscarinic receptor. So in order to understand what the anticholinergic does, you can't just say, oh, the anticholinergic is going to come here. He's going to squat its place, preventing the ACH from inhibiting, and then it's not going to permit the cell to do its metabolic function. No. To better understand what it does, you need to, in a first half, understand what the effects of acetylcholine does. And with that being said, that's where we start. So, ACH, acetylcholine, cannot pass through the membrane like a steroid can, like a liposoluble substance can. So, what does it have to do? It's going to have to activate a second messenger, and the way it does that, it attaches to its receptor, and what's going to happen in this case, there is a receptor here that is called a G stimulatory protein that gets turned on, right? And the way it gets turned on is that when it's off, it has GTP bound to it, meaning that it's off. And to turn the switch to on, when the ACH binds onto its muscarinic receptor, it will remove that GTP and will convert it into GDP. And that is what is going to turn our G stimulatory protein to on. And then the G stimulatory protein, think of it as Romeo and Juliet. Our G stimulatory protein is just going to circle the membrane, etc. It's going to go around the membrane until it finds our first intracellular enzyme, our first intracellular enzyme, which is, I'm just going to make a space here, which is a cyclase. Actually, no, it's not a cyclase. What am I saying? It's a lipase. So it's going to find its lipase, a type of lipase. And then what it's going to do, it's going to get switched off, right? So it's going to remove Once it's here, once our G stimulatory protein gets here, what's going to happen is that it's going to get inhibited or turned off. So it's going to remove its GDP and return into GTP. And then once the lipase is activated, what's going to happen is that here's what's where all the magic happens, because the lipase does everything. What the lipase does is that it's going to intake a bunch of molecules. So it's going to take a bunch of molecules, a lot of them, and it's going to break them down. Right? It's going to break them down. And then after it breaks them down, it's going to make other molecules. So there's two pathways here. So there's two pathways here. One of them is IP3. And one of them is DAG. DAG. Our IP3, and this is our, this is what, we're going to start with the IP3 pathway. The IP3 pathway is going to go all the way towards the and in the smooth muscles we don't have an endoplasmic reticulum. We have something called a sarcoplasmic reticulum. And that's our sarcoplasmic reticulum right here, rich in calcium. And so our molecular breakdown that just happened, which goes into two pathway, IP3 and DAG. IP3 goes in order to secrete, to stimulate the release, the high release of calcium out of the sarcoplasmic reticulum, because the smooth muscles, the sarcoplasmic reticulum in the smooth muscles, there's a lot of synthesis of calcium happening in there. And that's what it's going to go do. And once the calcium gets released, it's going to go do one thing. Actually, I shouldn't have done that. It's going to go do one thing. It's going to go bind on to an enzyme or a protein called calmodulin, creating the calcium / calmodulin complex. So it's going to create that complex. Whereas if we go to the DAG, this guy is going to activate another kinase. So it's going to activate a kinase. But they're both going to go do and have the same effects. What are these effects? Well, they're both going to go stimulate or augment metabolic functions. Metabolic functions. And these metabolic functions are going to either they're going to separate into two pathways, right? The first pathway is going to stimulate And this is our channel, our protein channel. This is a calcium protein channel. It's going to go stimulate the intake of calcium that exists outside. And in order to do the same result, so it's going to separate again into two separate parts. One of them is going to go directly do its final function, or it can either go secrete calcium inside of the cell from its channel protein by opening the calcium channel. And when you have calcium that gets inside of the cell, or because of the metabolic function, what happens is, first part we saw from the M3 receptor that, first of all, there's the glands over here. What the glands are going to do is you're going to have a high augmentation of liquid secretion. So an augmentation of liquid secretion, and whenever calcium intakes the cell, it binds on to the tropomyosin, myosin complex, etc., creating our contraction. So there is an augmentation of constriction. Whether it's bronchoconstriction, vasoconstriction, there is a constriction happening in the smooth muscles and the parietal cells that are present in the bronchi. So that's what our ACH does. Now, I'm going to draw another receptor here, another muscarinic receptor, and this is our anticholinergic receptor. And I forgot to draw the nucleus here of the cell. That's our nucleus right there. So we have our anticholinergic, and we have another acetylcholine here that just got secreted. Now you got your anticholinergic, right? He just loves squatting the house, the place, the spot of our acetylcholine. So acetylcholine will not be able to go to his place because it's being squatted by our anticholinergic. And who says anticholinergic also says inhibition of the ACH, which prevents it of doing its effect, meaning that if we take this all over again, you can now finally explain that the anticholinergic inhibits the ACH from activating its effect, meaning its second messenger system, activating the G-stimulatory protein that cannot activate its lipase in order to do molecular breakdown into two specific molecules. One of them that stimulates the secretion of calcium from the sarcoplasmic reticulum, the other one preventing it from activating its kinase in order to augmenting metabolic functions, which these metabolic functions will not be able to intake calcium in order, or to either go directly and do augment in order to augment liquid secretion or augment constriction. So it won't be able to do, there will not be this pathway involved, right? All right. This one was perhaps the longest one to explain. We're now going to go towards our adrenergic receptor. This one is the middle one. It's a lot more simpler to explain. But if we go back again to our if we go back to our graph over here, the pathway that interests us, you have our ANS going towards the SANS, our sympathetic part of the nervous system that gets controlled, which then will permit us to better understand what the adrenergic receptor is and its pathway. So we have alpha and beta pathway. I'm just going to remove this. So we have our beta pathway. So it goes here to here, stimulating the beta pathway, and then directly our beta 2 pathway, which we find a lot of beta 2 in the lungs. A lot of them are found in the lungs. And again, adrenergic receptors secrete adrenaline. Adrenergic receptors are it's basically adrenaline or noradrenaline or epinephrine, norepinephrine for you Americans. It's the same thing basically. So they act as a complement to these substances. So let's assume that there was adrenaline or noradrenaline that got secreted by our neurons. And then you take your salbutamol pump or your corticosteroid that has another beta 2, the beta 2 half of it, and you have your beta 2. Well, both of them are going to do the same effect. Again, the beta 2 act as a complement, so he can also connect to the same receptor, and they're both going to do stimulation. Again, since they're not liposoluble, they cannot pass through the membrane, so they have to use the second messenger system. So, but it activates another. It turns on a G-stimulatory protein, but this time, what's going to happen is that GTP, not GDP, but GTP keeps the G-stimulatory protein off. So what it's going to do, it's going to remove a phosphate, transforming it into GDP, which is going to turn this protein on. The protein is going to try to find its Juliet, so it's going to do, it's going to go along the plasma membrane until it finds, and this time it's not a lipase, but a cyclase. It's going to find a cyclase. So, right here we have our cyclase. And we have our cyclase right here. So what it's going to do, it's going to come and it's going to stimulate our cyclase, which is going to take, our ATP, which is energy, and it's going to convert it into cyclic AMP, a high amount of it. It's going to stimulate that secretion. And then our cyclic AMP is going to go stimulate or activate another kinase, but in this case it's called our kinase A. It's going to go activate a kinase A, which will, again, go do something called phosphorylation. Meaning that it's going to go put a phosphate on everything that it finds. Like Oprah, you know, it's going to give a phosphate to everything. So this guy gets a phosphate, the other guy gets a phosphate, everybody gets a phosphate, right? And now phosphorylation, what it's going to do is going to go do two things. One, it's going to go stimulate DNA transcription. So stimulate the transcription of DNA, augmenting it. Or it's going to go do another thing. It has another pathway I kind of forgot to draw our nucleus right here. Let's put a nucleus right here. So, it's going to go do transcription of DNA, or it's going to go stimulate metabolic functions. In this case, it's going to go stimulate proteins / enzymes. And on our case, it's mostly going to be enzymes. And these enzymes, one of them, is the inhibition of something called histamines. I'll explain what histamines are in a bit. And these histamines are going to give the result of this equals vasoconstriction of arteries but also the diminution of secretion of mucus, because histamines, what they do, they're secreted by special cells found in the lungs. They're going to secrete this substance called histamines. So if you inhibit that secretion, that the histamines cannot go into the blood plasma of the pulmonary arteries, and cannot be exchanged by the exocrine cells, the exocrine glands, in order to better secrete mucus, because histamines, that's what makes the mucus so viscous. It won't be able to do that. So with an inhibition of histamines, there's less mucus secretion, and also it's going to inhibit a certain protein or enzyme that does muscle contraction, and that's what creates our bronchodilation in the lungs. And another point is that there's a note. Notice how here we had the secretion of calcium, and here our kinase, our cyclic AMP went directly to secrete kinase A, and our kinase didn't even need to secrete calcium. So no no calcium secretion at all. So, if there's no calcium secretion at all, we can also proudly say that less calcium secretion also equals less constriction of the muscles. That's wonderful, right? And that's what we want. That's what we want for our patient. All right, now that we finally explained both of them, we can finally go to our combinate. So, our combination medication. And our combination medication is composed of two things. If you remember, one of them was our beta-2 agonistic long-acting / our corticosteroid. All right. What interests us is the corticosteroid, and this one is fairly simple. We know that if we do a little slash here and take out cortico and just leave steroid, we know that there's a steroid involved, meaning that it's liposoluble, meaning that if I redraw my cell over here with its actually, I'm going to make a bigger. cell membrane, nucleus with little holes. Oh, whoops, sorry. So like this, and this, and this. And now basically what our corticosteroid is going to do, we have our membrane over here. It's going to go act on our inflammatory substances secreting cells. And what are those, just by any chance? Well, who secretes inflammation? White blood cell. No, not white blood cells, but our white cells, the leukocytes. Leukocytes, macrophages, yeah, white blood cells basically are army defenders of the body. So we're going to go inhibit that with the intake of the corticosteroid. So as we explained, the first half, which is the a long-acting beta-2 agonist, is going to do the same effect as this, which is going to permit, in a second step, the corticosteroid to first, we have our cortico here that I will represent by this diamond shape right here. So this will pass directly through the membrane, and because it's liposoluble, and there's no mucus to grab onto because the beta did its effect, or the salbutamol did its effect, the beta did its effect, right? The corticoid can just go through, pass through the lungs, and pass through the cells, and get exchanged between the white blood cells, the leukocytes. So it goes inside of the leukocyte. It's right here. It passes through the membrane because it has diplomatic immunity to pass the border, because let's say it's a high-class politician. Unfortunately, it's stuck now. Why? Because the membrane, the intracellular liquid here, the intracellular liquid, it's liquid. So this guy over here is having a lot of trouble, because what he wants is to go inside of the nucleus, right? So he's going to have trouble moving in. So what you want is, which I'm going to represent by a heart, is you're going to have what we call, this guy over here is a G receptor. And this is our cortico stimulant. So this G receptor is kind of like a limo driver, right? He's gonna go pick up our little corticosteroid at the airport, because this guy, our corticosteroid, traveled by air because he got inhaled, passed through the membrane, and he gets picked up in his limo by this enzyme, this G receptor. They're both going to bind on to each other, and what's going to happen is that both of them are going to create a complex. And this complex is called the CSGR complex. Corticostimulant G receptor complex. And what's going to happen is that these guys are going to try, well, basically the driver, which is our G receptor, it's going to permit the corticosteroid to travel, and since it's liposoluble, again, it can travel through another membrane. And what is the nucleus? What is the nucleus made out of? It's membrane. It's a it's a phospholipid bilayer as well. It's as simple as that. So that means it can go through the border. Through the border. That's incredible. And then finally, once it's here, it's going to go target a specific part of the DNA. Let's say it goes and targets this part of the DNA. And then it's just going to squat over it. Like this. Like this. It's going to go squat on this part here. And it's going to modify the DNA transcription or the DNA, the protein synthesis, and it's going to do two things. One, it's going to diminish. So it's going to inhibit in order to diminish the secretion of pro-inflammatory substances by these cells. The second thing it's going to do, it's going to stimulate the secretion of anti-inflammatory substances. And it's as simple as that. That's what it's all going to do. That's what your corticosteroid is going to do, which is going to reduce the inflammation of our patient's symptoms. All right, we finally were able to explain the intracellular function of each type of medication. So now if we go back to our patient and finally answer his last question. Why all of these medications for the patient? In order to explain to the patient why he has all these medications. Well, sir, Mr. John Doe, you require all of these medication for your COPD. And note, COPD is a combination of two diseases, one of them being emphysema, which comes with another disease with chronic bronchitis. So, in order to ease your ailments of your diseases, the first part is your AAC, which is your long-lasting anticholinergic complements the beta and the beta. Our beta being the beta-2 agonistic short-acting medication, and the other one being our beta agonistic long-acting medication. We saw that it's going to complement both of these. So who says less constriction of either the muscles, meaning whether it's vasoconstriction or bronchoconstriction, it's just gonna be, there's just gonna be less constriction and less mucus secretion by the gland. So it's gonna complement that. It's gonna complement that. And then afterwards, the beta half of the combination medication of your Advair helps with the bronchodilation for a better intake of the corticosteroid. Why? Because the first part, as we mentioned, the beta is going to come and bronchodilate. Less dilation, so less constriction of the smooth muscles on the beta-2 receptor, and less secretion of histamines, meaning that there is less mucus, meaning that the corticosteroid can easily travel by air and do its effect inside of the inflammatory cells that causes inflammation. And then finally, also note that the salbutamol is prescribed in a just-in-case scenario. For example, it is only to administer only if the symptoms of your COPD are so out of control that your tiotropium and your corticosteroid combination are taking so much time to act that it's unbearable. In this case, your salbutamol, which is your inhalation, your beza, will be instantaneous. So that's what we can explain to the patient. Hopefully he understands why he needs all of these 3 medications in order to ease his ailments. So, with that being said, thank you very much, ladies and gentlemen. This concludes our live stream on the different types of respiratory medication. What we learn is that we have 3 of them: beta-2, anticholinergic, and corticosteroids, which are your main medication for respiratory therapy for patients suffering with respiratory diseases. Again, I'd like to point out the references. Thank you so much for Ninja Nerd Science and the RQESR. That's where I mostly got my information in order to do this live stream. Thank you so much for assisting to this live stream. You guys have been great. Hopefully I will do more live stream gaming videos and more science videos since I got out of school and I'm ready to go for more gaming and science live stream videos. So thank you very much for assisting for this science live stream of the year, of the end of the year. Please remember to subscribe, like, and share. I have been your post, AJP, and I will see you people next time.